Hello everybody. So good morning, good afternoon. Today we again coming back to see and to hear about TRANSFAC. So coffee break with TRANSFAC. Grab your coffee or tea like I did and relax. Also prepare your questions. Yeah, we especially coming here today, and generally these seminars about answering your questions about TRANSFAC, about tools which we are using, which you can use to analyze your data using TRANSFAC. And we collected several questions and grouped them together. And today is well, everything started from first question. Let me share the screen. Okay, so you see I have a list of questions there coming and I hope I will be able to answer them during my talks, during my coffee breaks. But still, I'm happy if you can give me questions just online. So for that, you put them into Q&A section here in the Zoom. And if I see there are the questions, I will interrupt perhaps and answer them. So you have priority questions. Still, I have several questions prepared, and so they were before. For instance, there was a question. I found TRANSFAC mentioned in publication that report about analysis of human data. Yeah, that's right. I am not working on human, but on Drosophila, and also sometimes on different plant species. Can I still use TRANSFAC? What are the limitations? Okay, so to answer this question, that's a very good question. Actually, of course, the answer is yes. You can analyze your non-human data using TRANSFAC. And let me explain, give a little bit of introduction to TRANSFAC again. Because not all of you perhaps were before in the similar seminars, and basically you are free to look at our recordings and of such seminars and coffee breaks which we did before, so you may find many useful answers to your questions and how to use TRANSFAC to analyze your data. So today, concerning the TRANSFAC in general, so let me open this famous tool. So the TRANSFAC, how it was created more than 30 years ago, and we still continue developing it. So basically, we are talking about transcription factor binding sites. And for instance, there is a transcription factor in human, NF-kappa-B, and which binds to binding sites in genome. And these binding sites are normally in promoters or enhancers of the different genes. And then this factor regulate the genes. It's switching them on or off depending on the conditions where this gene has to be working. And we are collecting the binding sites, known binding sites, which are experimentally proven by various experimental tools. And then we build a so-called positional weight matrix. Many people call it also motif or, just matrix. So actually, it's a representation of all these known sites in terms of such a matrix. So where we put certain weights to each nucleotide in each position of that on that motif. And when the nucleotide is big in that logo plot of that motif, it means this position is always occupied by a given nucleotide. And then once we build such a model, we call it model or positional weight matrix, we can apply it, and there are tools in the TRANSFAC associated with TRANSFAC. One of the tools which I will talk today is a match tool, and I will show it again for how to use it in different organisms, in different genomes. And basically this tool takes this matrix and scans it through the given nucleotide sequence of a new promoter or new genome, and it calculates in each position a score, which means how well this nucleotide sequence is fitting to that consensus, to that model. And if this score is higher to a given threshold, then we claim this site as a binding site for transcription factor. So another point, actually very important concept which we will talk today is a site enrichment. So actually, single sites very rarely regulating the genes. So you need many of those sites, and basically you can have many sites of the same factor in the promoter of the gene. You can have one site, but you can still have many sites. And also, if you have a set of genes which are doing a certain job, for instance, they belong to a certain pathway, and we talk about these pathways today, or they are simply switched on altogether in particular conditions. So they are, for instance, upregulated upon stimulation or upon infection of that organism. Then the promoters of those genes, they are enriched by the motifs of that of particular transcription factors. So if they're enriched, we claim that this factor is playing a role to regulate the gene. So actually, it's a way how to find out which factor is important, because we simply look which motifs are enriched. So, and for enrichment analysis, we take sets of promoters of our interest, or a set of regions of our interest, and then we compute how many binding sites which are higher a particular threshold could be found here in this set of promoters which are regulated in a particular way, and compare, and we call this set of promoters yes sequences, and we compare them to a background. And background could be either a whole genome background or a background just artificially created random sequences, or a real background, for instance, promoters of genes which actually didn't change expression in the same condition. And then if you can, as you can see here, if you see a lot of sites found in this set, but not in this set, and this can be how big is the difference, and whether it's statistically significant can be estimated by a certain formula, by binomial distribution. And we can say that this set of sequences, or this pathway, is enriched by that particular motif. That's important concept, and it actually helps to identify individual transcription factors which are responsible, or which are supposed to be responsible to regulate a particular set of genes. Another concept which we'll talk today, and which is very important, is actually a concept of site combinations. So, that's something currently becoming very clear, that individual transcription factors, they are regulating many genes. And if you are talking about very specific regulation of a very particular set of genes, which should be switched in complex situations, then this is regulated by complexes of factors which interact to each other, which bind to that region, which have many sites for different factors. And we call this combinations of sites and factors. We call them composite elements, or composite modules. So, we have to search for these composite modules in the regulatory regions of genes in order to understand how these genes are regulated in really very particular manner. And how it is working, actually, recently it became very clear and even evident how this works. And actually since become even very much unclear at the end, because we are talking here about very fuzzy interactions of factors are not really firm bound to their binding sites. They are interacting to each other. They form so-called phase separation. For that, I recorded a video. I want to show very briefly to you today. This video, which shows basically this concept, how the transcription factors interact to each other, and what are the physical physical needs to understand how they interact with each other. Let me switch on that video. Just a moment. So I will. I have it here on YouTube. And by the way, we have a number of videos recorded on our website. You can watch them. We have a way explaining how to use our tools, what things means, what means all these transcription factors, why should you find them, and so on. So and just watch this video. Just let me start it.
So let's go back to our beloved transcription factors. Here they are. So these funny guys. And when the cell is developing or growing, there are many factors which are active in the cell. And as we know, they are bound to different places in genome, so-called regulatory regions of genes, either enhancers or proximal promoters. And what is interesting, they are binding in cooperative manner. What is it? It is important to understand the nature of this cooperative binding. Recently, it was shown that the cooperative binding of transcription factors is going on according to the so-called phase separation model, which you can see in the glass of water. Let's take a glass of water and put in it a little bit of oil. And you can see those droplets of the oil are quickly start binding to each other like transcription factors would do. And finally, those liquids are phase separated. In the nucleus of the living cell, this nucleation of the binding is actually promoted by the DNA sequence of the enhancer region where those transcription factors are actually attracted. And if this region has a mutation, which actually destroys the binding of those transcription factors, then this nucleation is not happening and formation is dissolved. But what happens when there are many different transcription factors in the cell? So like in this picture, which you see here, actually they found that it's the same thing. So those different transcription factors, they can organize together, they can bind together in forms so-called condensates, which are either homogeneous or heterogeneous. So they can consist of several transcription factors acting together. And recently, it was demonstrated that there's conglomerates, those concentrates of different transcription factors can be seen in the microscope. They actually can see them as a physically observed droplets in the cell. So
as you can see, a little bit of introduction, that things are really complicated. So on one side, but also simple. So various transcription factors, to regulate their genes, they really have to interact with each other to form these condensates, like the condensates you see on the glass of condensates of water. So here there are condensates of transcription factors interacting to each other. So and since we have to actually try to find these combinations, and this one example of these combinations when simply pairs, when one transcription factor is bound to DNA and it attracts another transcription factor by protein-protein interaction and actually recruited to bind to DNA, although the binding of this factor by itself would be never happening. So that compensates the energy loss when there is a poor binding sites, which not very thin. And in general, we have as I explained also before, but just a little bit very shortly here, we have a tool, we have an algorithm inside our platform, which uses genetic algorithm in order to find combinations of transcription factor binding sites, and through that actually to understand which transcription factors are really having master role in the regulating our set of genes. Okay, so that's for a small introduction. I think it's helpful. So coming back to the original question, so the question was, of course, we have many tools in TRANSFAC to analyze human genome, human genes, mouse genes, rat genes. So those which are well studied, and we have a lot of tools. We have actually also a big, workflows like Genome Enhancer. You should try it, which work just for human data, unfortunately, for those who work not with human. So, and that Genome Enhancer does everything. So automatically, it analyzes promoters, it predicts combination of sites, goes to pathways, to networks, find drugs, so does everything. So you can listen about that in my previous seminars. But what about those people who analyze model organisms? Those guys, you know, who are not so fortunate to work with a human mouse or rat. Or maybe actually they are really fortunate because your guys are working in terra incognita, so when not so much data are available. So, and we started from many years ago to collect various matrices in TRANSFAC from various species. And as you can see, actually, this is a logarithmic scale. So it means different species have a very different number of matrices in TRANSFAC database. So that's TRANSFAC. And of course, you see like Homo sapiens and mouse and rat have the maximum number of matrices. But we have also actually quite high number of matrices, for instance, for Arabidopsis. We have actually for Drosophila, also for fungi. Fungi, of course, that's mainly yeast, which are considered, but not only. Few are for smaller species as well. Not so well studied. So you see, there's several kind of not very big set of organisms, but quite number. And I must say that TRANSFAC in that respect is quite good. So, for instance, plant matrices, which we will talk today about. So they not only Arabidopsis, but also maize and tobacco, tomato, wheat, also barley, apple. So some few matrices, yes, have from different plant species. Actually, since this distribution of matrices between different species is so big, and not only because of that, but also because of that, when we analyze data from a particular species, for instance, when we analyze data from human, we use matrices, not just human matrices. We use matrices which were constructed on the basis of transcription factor binding sites collected in similar organisms. For instance, we use matrices constructed in mouse and rat, and also in other mammals. And that's reasonable because transcription factors, although these proteins, they evolve very rapidly, but in fact, the DNA binding domains, so the domain of binding to DNA, which defines what is a motif of binding, is often very conserved. And actually, it makes sense to combine matrices from different species which belong to the same, to big groups. So we have vertebrate matrices for all vertebrates. We have plant matrices for all plants, insects. So we have big groups, and they have even in the matrix nomenclature, they have this first letter, which defines this big group. And if you analyze, for instance, a plant, some new plant, and we look at this today, you can use all matrices from plants, and this will generate hypothesis. Even a transcription factor, which was used to build such a matrix, comes from a different species. But as I said, since DNA binding domain may be quite unconserved, you may create a hypothesis that similar protein, similar transcription factor in that particular species which we study may also bind to that place, and also may interact with promoter and regulate the genes. All right, so that's about it. So other pictures maybe I will show later when explaining other questions, but now I will go online and show how to use TRANSFAC for different species. So for that, we will go to our website. So that's our website where you can log into our system to analyze your data, to put your data, and to apply TRANSFAC. So for that, we log into system, and I have here my login. So if you have an account in our system, you have your own login. So you in this particular login has a TRANSFAC, a subscription, and also proteome. So which means we have information about various species, not only human, mouse, and rat, but many different other organisms. And we have, so this subscription is connected to geneXplain platform, which we are now going to start. So as an example, I used several examples today. And by the way, again, reminding you, if you have questions, just don't hesitate, write questions in Q&A. While I'm talking about everything, I'm just talking, I may stop and explain a bit more, some things which you have questions about. But okay, we'll have also enough time at the end to answer your questions. But let me first proceed with that, what I prepared. So first example which I prepared actually is on Drosophila. So, you know, Drosophila is quite a well-known model organisms. It's maybe one of the most, oldest genetic model organism. Fruit fly, and Drosophila is actually studied very well. And there are a number of data available for Drosophila. For instance, I selected this data from Gene Expression Omnibus. It's a combination of RNA-seq and actually ChIP-seq to study a very interesting phenomenon. Actually, that's related to what I was talking today in the beginning about all these fuzzy interactions of transcription factors to their binding sites and all this phase separation model and everything. What I mean is that very often there is a question, okay, I have my transcription factor, tell me what is a list of target genes for this transcription factor. And that's in fact sounds like a simple question, but in fact it's very complicated question, and in fact it's not also a correct question, because transcription factor may have many different targets depending on the conditions in which you want to analyze. So in which tissue, in which cell line, under which circumstances different genes would respond to a particular transcription factor. So that's moreover, actually, if in this experiment, which is present here in this paper, in recent paper, what they did, they took a human transcription factor and put it as a construct and expressed it in Drosophila. And you know, the Drosophila genes have never seen this factor in their life, so in their evolution. So it's very new protein coming into the system. And what is a surprise that this transcription factor, the human transcription factor, start regulating Drosophila genes. So which means for transcription factor, it doesn't matter. So it just sees the target, it binds to it, and if there's other factors which can interact with this factor, the endogenous factors, they may found this phase separation, they got activated, and the gene is activated. So that's what they show. They actually made a ChIP-seq experiment and found that this transcription factor is actually bound to many places in the Drosophila genome. And also they made RNA-seq experiment, and they found that upon this binding, a number of genes are heavily upregulated, and also down-regulated. So, and I was interested to see where these bindings are happening in genome, whether actually Drosophila genome, that regions have a target, have a motive for that factor. What is a combination of these other endogenous factors which make this regulation actually happening. And so for that, I just want to briefly show this one slide where I presented this, this kind of experiment. And that's very often the case when you have two types of omics data from the same system. So you have transcriptome, for instance, RNA-seq, so you know which genes are expressed, differentially expressed, and you have ChIP-seq or ExaSeq or ATAC-seq or any other method which detects actually which regions of genome are active. So in whatever sense. So in this particular experiment, they were interested in ChIP-seq, which means they were studying which regions of genome are bound by that transcription factor. But if it's ATAC-seq, for instance, it just shows which regions of genome are opened and ready for regulation. So, and then many factors can bind to that. So, and by combining these two, our system can actually do the following. What we do here, we take a transcriptome, from transcriptome, we take those genes, we take the upregulated upstream regions of those genes, or maybe also including downstream, including introns, depending on our task. And then we take ChIP-seq data, and which shows us the peaks where the factor is bound. And then we intersect these two things, and we identify those regions which on one side are bound by this transcription factor, but in another side, they are in the vicinity of a start of transcription of the upregulated gene. So these two lines of evidences gives that it's not just bound there and doing nothing, but probably bound and upregulating those genes. And then I ask a question, okay, we have this factor bound there, but what other factors, endogenous factors in Drosophila can be combined with this factor together and regulate this gene? So we can find the cofactors which help this particular factor to upregulate, to drive the genes, or maybe other cofactors which can drive the genes down-regulate the genes. So depending on the cofactors, the same transcription factor can either upregulate the genes or down-regulate the genes. Okay, so that's on the schema. And now if we go back to our tools, so I'm still have to open my okay, so here this example. So what I did, I took this data from that paper I just showed to you. And by the way, if you haven't seen before, this is a so-called geneXplain platform, which has a number of pipelines, so like hundreds of pipelines. So, but today we have very focused interest, and we'll talk about a couple of particular pipelines. So what we see here is are two things. One thing, I uploaded the peaks. So the peaks are actually, let me just click on them. So you see this is opened in the Drosophila genome, and these are the peaks. So in the genome, so we can, the genome has several chromosomes. Also mitochondrial genome is here, so those peaks are located in different regions of genome. So what are the peaks? So ChIP-seq peaks, they in the center or maybe somewhere there's a factor was bound, but then using the sequencing technology, these authors were able to identify the region where those factors are bound. Now, what we can do, we can add here the genes, information about the genes. So let's put here these track genes, and that's what what I have prepared. Actually, for instance, this peak is located near start of transcription of that gene, and so these are the genes, and all genes in genome, I mean all Drosophila genes, but we have a set of genes which are up and down regulated. So that was also that's what I uploaded from that paper from Gene Expression Omnibus. How I upload this data I was showing last time, so you may look at this coffee breaks recordings and see the details how to load different types of data. So actually the system automatically recognizes what type of data you are loading, and depending on that it either creates a track or just a list of genes. So for instance, this is a list of 366 genes which are upregulated in the Drosophila in the condition when this human transcription factor was introduced expressed in that system. So it started to upregulate genes, and you see actually some of the genes, for instance, this one is really heavily upregulated. It's by the way, it's HSP, so it's you have to probably do a lot of interactions with the new protein, with this intruder. So the protein just came in that system. So this gene has heavily upregulated. So it's log2 is 8, which means it's many times higher expressed than in the control Drosophila. And now this is 366 genes, but now I want to intersect these two sets of genes, these two types of data. So I want to intersect genes which are upregulated with all peaks in genome which were found to be bound by that factor. How to do this intersection? So for that, first, what I did is actually I converted this into a track. So there is there is a let me see. Okay, what I did, in fact, I did a diagram. So so let me just show to you how to do that. So here in the analysis tab, we have function which is called, we have many functions, but the function which I was using was is called track to we have to gene to track. We have a gene set and we want to convert it to track. What do I mean? So we have just a list of genes, right? But now I want to put them into genome and to know where those genes are located. So these particular 366 genes. And for that, we have this gene set to track function. So you double click on it, you drag and drop here the gene list. And what you do, you actually specify what track you want to see. So I want to see actually a big distance from start of transcription. Let me write 5,000 nucleotide and perhaps 1,000 nucleotides downstream from TSS. So there's a TSS, start of transcription of the gene, and I take a very long upstream region, 5 kb, and 1 kb downstream from TSS. Why? Because in any of that region, the transcription factor, this new one, this human one, can bind, and we can see can be very long distance interactions with that gene. So such enhancer could, new enhancer could be pretty far away. So for instance, 5 kb or even higher. So we created such a track. So I will not. So we basically. So yeah, let me run it. So that's a track. So you see what this track, how does it look like? So for instance, we have this gene, which is obviously was upregulated, and this is a track which is near start of transcription of that gene. So 5 kb upstream and 1 kb downstream. And by the way, this peak, these peaks, right? So one of that peak is located just in that region. Okay, so that's one of the candidate for really for a region which is not only bound by this factor but also regulating my gene. Good. So now what I want to know to do is would like to basically see all the cases when my peaks are intersecting with my genes. And I did that by Venn diagram. And so here you see the result of that Venn diagram. So you see these upregulated genes, 366. And here the downregulated genes, 392. And we have here all these peaks, all the genes which have at least one peak. So actually, I can show you how they look like, these genes which have at least one peak. It looks like that. So this is done with another function which convert track into the gene. It actually put the tracks on the genes which are nearby. And what you see here, you see these 12,000 genes, and actually which have at least one peak somewhere inside this gene, and plus minus 5 kb from the upstream region of the gene and downstream region of the gene. So this each of the red bar here is one peak where this transcription factor was bound. And actually, what we can select here, we are interested only in those genes which have at least one peak in the 5' region of the gene. So you see these are the genes which have most of the peaks in the 5' region. So which means this may be the genes and the situations when the peaks when those regions are really regulating expression of those genes. All right. So then by just intersecting these genes with those peaks, I got this diagram as which I showed to you before. So, and which means that if we look at the upregulated genes in 291 cases, we have at least one peak in 5' region of that gene. So we have to take those peaks and analyze their sites in the peaks. All right. So, all right. Basically, by doing these various intersections, I will not do now this online, just to show you that we have this, intersect filter one track by another function, which means I take track of peaks and filter them by track of promoters. And at the end, I got this this set of peaks, which are here, up peaks in 5' region of promoters. We can drag and drop them here. And indeed, this is only those peaks which are intersecting with promoters of upregulated regions, upstream regions, sorry, of upregulated genes. So for instance, this peak, which was very far away from that gene or from any gene, is not included in that list. So we have this list of actually, we can have a look how many of them are there. So it's 329 peaks which are located in promoters of upregulated genes. All right. So and then to analyze these peaks, you have to go to this ChIP-seq region of area of our platform, and here we have analyze ChIP-seq peaks. And here we have search sites in for transcription factors, and we have various versions of those algorithms. And I applied here the algorithm which is the latest version, which uses TRANSFAC. So what you do, you simply drag and drop here your peaks, you select your species, and in this case it's Drosophila, and you have to specify which positional weight matrices, which motifs you want to apply. And here, one condition is you can simply click here and go to to insects and select all insect positional matrices from all insects. But of course, insects are very much different. And as you remember, particularly for Drosophila, we have a very good collection of positional weight matrices. They're approximately half of the insect matrices, which are around 700, half of them, around 300 or 400, are coming from Drosophila. So actually, these particular positional weight matrices are here. This profile. So let me simply close this. So this profile contains 365 positional weight matrices from Drosophila. And so I prepared this profile, which means when I start searching for the binding sites, I have to take this profile instead of standard profile. So then only these 360 matrices will be used. And then what happens? So basically, I analyze that, and here you see I have many different results. Let me show you one of the latest. So I got this set of matrices, which are actually, as you remember, we have this concept of enrichment. So these matrices are enriched in the peaks, in the regions which are bound by that human transcription factor. So additional motifs for endogenous transcription factors are also enriched in those peaks. So these are the candidates for the cofactors, which make this human factor actually functional, to make them this human factor to regulate the genes. So, and it's quite a lot, so 126 different positional weight matrices. And here, this yes to no ratio tells you how much is this different. So how much is an enrichment? So 8 times or 5 times these sites are more frequent in the regions, in the peaks, compared to the background regions. And this is a p-value, which is significant. For instance, this factor is 5 times higher and also very much significantly enriched. So, and but then, as you say, to find really the master transcription factors, not all these 126, but really those which are in combinations can work together. I run here this Composite Module Analyst, CMA, so this genetic algorithm, and it returns this result. So let me open it. So let me first close this. All right. So this is finally these 290 peaks, which are located in promoters of upregulated genes. These are peaks where, this is a region of a peak. So peak can be very long. It's up to 6,000 nucleotides. The peak can be, by the way, we can look at the peaks itself. So just as if you open it as a table and look at the size of the peak can be 200 nucleotides or up to 6,000 nucleotides. Can be very big peak. So so that's why this region can actually cover not only that gene, but can also intersect with other genes nearby. But what we can see here, you see clear clusters of the transcription factor binding sites, which were found by our algorithm. So clusters of these transcription factors. And now, so what we can see here, for instance, by the way, we can see here also information about how good is discrimination of those clusters in these peaks compared to the background regions in the Drosophila genome. Background, actually, not really background, but not any background, but background in near transcription start sites. So really promoter background, but these are other promoters, not those ones which are regulated by this transcription factor. So we got that discrimination, and we got these transcription factors, the motifs, as key factors regulating this gene together with the humans. But now actually one last thing to show that, in fact, if you look at this, by the way, we can put here these sites. So we drag and drop here, and you know these are the sites for these cofactors. All right, and we can zoom in, zoom out. But what is interesting, we can also look for sites which are found by the matrix from the human by the human matrix for the transcription factor from another organism. And here I found those sites. I think they're here, so we can click and see. So for instance, here you see this peak. In this peak we have two sites for this human factor. Pretty good sites actually. The score is 0.93, and which are accompanied by other sites from the endogenous factors, which in our opinion help to regulate those genes. Okay, so that's the Drosophila story, but as you remember, I promised to tell something about palm, and that's a little bit different story. Let me just go quickly to that. You know, actually, that's if you look at our databases which are connected in our platform, we see these model organisms, like you see this Arabidopsis, the Drosophila, you know, of course, human mouse rat, nematode, Saccharomyces, zebrafish. So that's the most important model organism. But as many people are working with other species, and then another question: can we actually use other genomes for analyzing transcription factor binding sites using TRANSFAC? The answer is yes, and that's a little bit more complicated. So there are no ready pipelines for that. So for that, you have to first upload the whole genome. And for that, very useful, there are many resources which can be useful. For instance, I am interested in palm genome. Some of my friends were involved in sequencing the palm genome. So let me open that. Yes, actually. All right, so that paper, which was published by my colleagues, Tatiana Tatarinova, and where actually Viktor Solovyov, which is my friend, was involved in predicting genes in that genome. Actually, this genome is published, so as a draft, and you have to download it first into the geneXplain platform, and then analyze it using our tools. So I will show now the result of that analysis. So let me go to that. Actually, genome. So you see this palm genome. Since we don't have this among our standard genomes, first you have to download this genome into the platform. And for downloading, actually, there are a number of websites where you can get genome, for instance, for first of all NCBI, but actually for palm genome, they're very useful. Resource is a special website from Malaysia, and which I'm using to download this genome. And then this genome is distributed as FASTA file with several reads or scaffolds in that FASTA file. So you simply upload it into the system, and it's immediately open here in this genome browser as a new genome. So you see in palm, actually, there are standard chromosomes, but there are a number of contigs which are not yet associated with any chromosome. So at least in that draft genome which I'm using right now, so there are many advances in the last years done into completion of that genome. But even if genome is not complete, so just having scaffolds is enough to start analyzing promoters of the genes. For that, you need actually genes. And to get the genes, actually you have to have a GTF file, so a gene transfer file, which you can also upload into the system, and you can drag and drop this. I'm not sure if it's, no, that's not the right one. So I have, there are several annotations of a palm genome. I'm just in the process of doing that. So, and then once you have such a gene list, so here what I did, I tried to get promoters of the genes which are responsible for resistance of palm to different, bacteria or infections. So there are so-called resistant genes which were published in that paper, and applying the same strategy, basically by applying the search sites in the promoters of resistance genes, and then analyzing compositions of those sites together, I came up with these results, and these are the actually the key factors, the master regulatory transcription factors in Drosophila, well, in palm, I'm sorry, which are regulating resistance genes. So they are located in promoters of those genes, and among them, for instance, there is a GATA factor. It's this GATA factor. Of course, we don't have specific palm positional weight matrices. They are not yet available. That's why we are using all collection of plant positional weight matrices from TRANSFAC, and this particular matrix is about GATA factor, which is GATA is also known in family of factors, also known in human. So it's a very distant organism, but still this transcription factor family is pretty conserved. And here we have this factor also in plants. And obviously we found here, for instance, in the promoter of those genes which are responsible for resistance of the palm to infection, and there are a number of bad infections which are affecting palm trees, and with that we see which transcription factors are playing a key role in regulating such genes. So that's very brief information about analysis of genomes which are not yet in our system. So if they are not yet among our genomes in the system, no problem. Of course, the prepared pipelines will not work, but with using different tools in our platform and uploading genome, uploading the gene annotations, and then analyzing promoters, you can get the key transcription factors regulating your genes in model organisms or in novel organisms. Okay, so with that, I want to stop my big speech, and I'm waiting for your questions. So, by the way, here is a track of genes, I guess. Is it? All right, yes. So these are actually the genes, which are resistant genes found in that publication. And then in the promoter of that gene, as you see, we found the cluster of transcription factor binding sites. Let me go to my previous questions. All right, so there's this question: how can I find master transcription factor that regulates specific pathways in Arabidopsis? Yes, that's a very interesting question, actually. Where to get pathways, first of all, and that's something I can show, but let me first look at your questions. There were some questions. You mentioned about the standard genomes in your system. What are they? All right, so again, I will show to you. So here in the databases, in the geneXplain platform, you see these genomes which we have in the system. So among them, so you see this ensemble where you have ensemble right in front of it. So we have Arabidopsis, we have Drosophila, then several genomes from human, several actually releases of genome. We keep them with certain time delay for a certain time, because some results are done by some users maybe using old genomes, and they have to be still in the system. So we have mouse and rat, we have nematode, you see. We have Saccharomyces cerevisiae and this yeast pombe with a very complicated name. So 2 species of yeast. And we have a zebrafish. So these are the genomes which we have in our system. And other genomes, as I said, you can also use, but for that you have to upload first the genome. All right. So, okay, still waiting for your questions. So concerning the pathways, for instance, for Arabidopsis. So the pathways, the best, I would say in my experience, the best database for pathways for many organisms is still ChIP-seq. So for instance, in ChIP-seq, I looked recently for various pathways, and for instance, this is the big pathway for photosynthesis for Arabidopsis. And so what is good here, it's also showing the list of all genes which are working in that pathway. So you can download this list and upload it into our system, and that will be a list of genes which are regulated, which are working in that pathway. And then, of course, this exactly how the question was formulated. Can we find actually a master regulators transcription factor which regulate genes in the whole pathway? Indeed, for the pathways, these genes has to be coordinately regulated. And because it's very bad if one gene is overexpressed and another is not in the same pathway, so things will not work. So you have to coordinate expression of the genes. That's why very often promoters of genes belonging to one metabolic pathway or one pathway like this big one, which works in the photosynthesis, they have similar binding sites for transcription factors. And I analyzed, for instance, for Arabidopsis, I analyzed promoters of pathways of glycolysis, and I'm now analyzing also pathways for this photosynthesis pathway to find the combinations of sites and to find the master transcription factors which regulate this pathway. All right. So there are repeating questions about which profile to use. All right, so actually I spent a bit of time in my first coffee break, maybe 2 months before, where I was describing various profiles and what does it mean. Just briefly repeating here again. So in TRANSFAC database, we have this concept of profiles. So if you go to TRANSFAC here in this TRANSFAC database, profile is simply a collection of matrices. And so if you look at the profile section of the database, you have a number of profiles, and maybe for some people it can be too much, of different profiles and not easy to select which one to use. So, and first of all, as I mentioned today, there are profiles which contains matrices from different big classes of organisms, like insects I mentioned, or fungi, for instance. If you click, you see these profiles of 367 matrices from fungi, so from different classes, a collection of matrices. And from vertebrates, we have, of course, also one of the biggest collection. For instance, here you have 7,800 matrices. So for vertebrates, and all of them can be applied to analyze, for instance, human genome. And they have these different profiles. They have different kind of extension of their name, which actually explains how they are calculated and what are the cutoffs. So, for instance, here you see these are cutoffs, very important parameters for each matrix, which tells at which level we consider this matrix as significant, this binding site is significant or not. So, and these were two cutoffs, cutoff for the whole matrix and cutoff for the core of the matrix. And these cutoffs are computed based on the different average frequency of nucleotides and denucleotides. And for instance, in this case, they were computed allowing that in a random human genome, basically the binding frequency of each matrix would be 1 hit per 1,000 nucleotides. So these cutoffs are then kind of guarantees that at least once per 1,000 nucleotides, you will see in the average the binding site for this factor. So, and there's another cutoffs and so on. So there are more explanations how we calculated this min FN, min FP, false negative, false positive cutoffs. So please have a look at those videos. All right, so I think we should finish for today. So there are also some many other questions coming every day. Sometimes pretty funny questions. For instance, there's a question: what is the most important transcription factor in bacteria? You know, in these bacterial circular genomes, and I must say there's probably one most important factor, which is called what is the abbreviation of that actually? So I have to look. So, but in general, there are not such a factor which can be most important transcription factor for the whole genome. For instance, although in bacteria there may be fewer factors than in eukaryotic genomes, but still they all play a very important role in various processes. And if there would be one factor, which plays the most important role, we could call it like Lord of the Rings, because bacteria are the rings. So that's just, very, sometimes we get some funny questions. So if your question is funny, also send it to us. We can answer it as well. Thank you for your attention for today. And you will see these recordings of that seminar. If you need some more details, you can watch it in those recordings or just send us more questions and we will be happy to answer. Okay, goodbye.